Hello student, I am Professor Devashish Bose, Head Department of Criminology and Forensic Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. Today, I am going to present a lecture of BSc 5th Sem, Forensic Toxicology on the unit Metabolism of Drugs, which has been jointly prepared by myself and Miss Neha Sharma, PhD scholar, a UGC JRF at Department of Criminology and Forensic Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar. Let's start our discussion while taking a look at what we are going to learn today. Today, we will see module number 1, Introduction, module number 2, Absorption, module number 3, Distribution, module number 4, Biotransformation or Metabolism, module number 5, Excretion of Drugs, and finally module number 6, Conclusion. Let's start with module number 1, Introduction. In previous lecture, we studied what is drug and how it can be administered in the body. We also understood about the action and response of the drug. But after administration and before the response comes, some other mechanism also happens to that particular drug, that is distribution, transformation, metabolism and excretion of drug. These all phenomenon we will study under the heading pharmacokinetics. So, pharmacokinetic is the quantitative study of the drug movement in, through and out of the body. Pharmacokinetic considerations therefore determine the route of administration, its dose, latency of onset, time of peak action, duration of action, frequency of administration of drug, etc. All the pharmacokinetic process involve transportation of the drug across biological membranes. Drugs are transported across the membranes by passive diffusion and specialized transportation. Passive diffusion. In this diffusion, there is not any active role of membrane. Drug diffuses in the direction of the concentration gradient. This is the most important mechanism of the drug because drugs are foreign substance and body developed special mechanism for normal metabolite. Lipid soluble drugs diffuse by dissolving in the lipoid matrix of the membrane, the rate of transportation being proportional to the lipid water partition coefficient of the drug. Greater the difference in the concentration of the drug on the two sides of the membrane, faster is the diffusion. Filtration is another transportation passage of drug through aqueous pores in the membrane or through paracellular spaces. This can be accelerated if hydrodynamic flow of the solvent occurring under osmotic pressure gradient. Lipid insoluble drugs crosses biological membranes by filtration if their molecular size is smaller than the size of the pore. Majority of the cell have small pores and most of the drugs are not able to penetrate it. However, capillaries have larger pores and most drug can filter through these. Now we will look at the specialized transport. Specialized transport can be carrier mediated or by pinocytosis. Carrier transport. The drug combines with the carrier present in the membrane and the complex then translocate from one phase of membrane to other. Carrier transport is specific, saturable and competitively inhibited by analogues which utilize the same carrier. This can be of two types. Active transport. The transport needs energy because movement occurs against the concentration gradient. It results selective accumulation of drug on one side of the membrane. Then is facilitated diffusion. It translocates even non-diffusible substances but along their concentration gradient, that is no need of energy. This is more rapid than the simple diffusion. Now we see pinocytosis. Transport by this formation of vesicle across the cell is known as pinocytosis. Every one of you know about this. This is applicable to protein and other big molecules. After the topic of transportation, now we move to the next step, that is absorption. Now we will discuss how these drugs are absorbed in the body. So, coming to module number 2, absorption. Absorption is the movement of the drug from its site of administration into the blood circulation. Rate of absorption is as important as the fraction of administered drug that has been absorbed. Many factors influence the absorption of drug. That is aqueous solubility. A drug given in watery solution is absorbed more quickly than solid form. Drug given in solid form must be dissolved in the aqueous biophase. Rate of absorption of poorly water soluble drug is governed by its dissociation constant. Concentration. Passive transport is governed by concentration gradient. Therefore, more concentrated drug absorbs rapidly. Area of absorption surface. Absorption rate is proportional to the surface area. That is, more the surface area, rapid will be the absorption. Vascularity of absorbing surface. Blood circulation removes the drug from the site of its absorption and maintains concentration gradient. Increased blood flow hastens or fastens the absorption of drug. Route of administration. Injected drug is absorbed more quickly than if the drug is taken orally, because orally drugs have been to be passed the lipodial epithelium of the gastrointestinal tract. If the drug is less lipid soluble, then absorption will be less. Presence of food also plays an abstention for the drug and retards the absorption. Apart from this, some drugs also get degraded in the gastrointestinal tract. If the drug is administration through intact skin, the absorption will be least. Then we come to other form of injection, that is intraarterial, intravenous, intramuscular, prenatal routes. Intact skin. Here I want to discuss about the bioavailability of the drug. Bioavailability of the drug refers to the rate and extent of absorption of the drug from a dosage form as determined by the concentration time curve in the blood or by its excretion in urine. It is a measure of the fraction of administered dose of drug that reaches the systemic circulation in the unchanged form. That is, bioavailability of drug when injected intravenous is 100%, but it will be lower in case of oral administration because the drug may be incompletely absorbed, or the absorbed drug may undergo first pass mechanism in intestine wall or liver or be excreted through bile. However, for a large number of drugs, bioavailability differences are negligible. Now we come to module number 3, that is distribution. You have learned about absorption of drug. Now, what happened after absorption? A drug enters or passes through the various body fluid compartments such as plasma, interstitial fluid, transcellular fluid and cellular fluid. The distribution of drug depends on its lipid solubility. Again, I am mentioning it. Ionization at physiological pH, extent of binding to plasma and tissue protein, and regional blood flow. Once a drug reaches to bloodstream, it gets distributed to other tissue that initially have no drug. If distributed according to concentration gradient from plasma to tissue, movement of drug proceeds until an equilibrium is established between the unbound drug in plasma and tissue fluid. Apparent volume of distribution. It is defined as the volume into which total amount of drug in the body would have to be uniformly distributed to provide the concentration of the drug actually measured in plasma. Presuming that the body behaves as a single homogeneous compartment with volume V, into which drug gets immediately and uniformly distributed. Therefore, V is equal to dose administered intravenous. Plasma concentration, it can also be defined as the volume that would accommodate the entire drug in the body if the concentration was same throughout as in plasma. Redistribution of drug. Highly lipid soluble drugs given intravenously or by inhalation initially gets distributed to organ with high blood flow. Example, brain, heart, kidney, etc. Later, less vascular but more bulky tissues, that is muscles, fat, take up the drug. Plasma concentration falls and the drug is withdrawn from these sites. Greatly is the lipid solubility of the drug, faster is its redistribution. Blood-brain barrier. The capillary endothelial cells in the brain have tight junction covered by neural tissue constitute blood-brain barrier. A specialized system not only maintains brain fluid composition constant but also protects brain from toxic material. A similar blood CSF barrier is located in the cordial plexus. Capillaries are lined by cordial epithelium having tight junctions. Both these barriers are lipodial and limits the entry of non-lipid soluble drugs. Therefore, only lipid soluble drugs are available to penetrate and have action on CNS, that is central nervous system. The blood-brain barrier is deficient at the CTZ, in the medulla oblongata and at certain preventricular site, anterior hypothalamus. Exit of drug from the CSF and brain. However, it is not dependent on lipid solubility and is rather unrestricted. Now, after finishing module 3, we come to module 4, that is biotransformation, or more easily metabolism. Metabolism is the enzymatic conversion of one chemical compound into another. Most drug metabolism occurs in the liver, although some process occurs in the gut wall, lungs and blood plasma also. Overall, metabolic process will convert the drug into a more water-soluble compound by increasing its polarity. The liver is the principal site of drug metabolism, although metabolism typically inactivates the drug. Some drugs' metabolites are pharmacologically active, sometimes even more so than the parent compound. An inactive or weakly active substance that has an active metabolite is called a prodrug, especially if designed to deliver the active metabolite more effectively. Drug can be metabolized by oxidation, reduction, hydrolysis, hydration, conjugation, condensation or isomerization. Whatever the process, the goal is to make the drug easier to excrete. The enzymes involved in metabolism are present in many tissues but generally are more concentrated in the liver. Drug metabolism rates vary from person to person. Some persons metabolize a drug so rapidly that therapeutically effective blood and tissue concentration are not reached. Another metabolism may be so slow that usual dose have toxic effects. Individual drug metabolism rates are influenced by genetic factor, coexisting disorders and drug interaction. For many drugs, metabolism occurs in second phase. Phase 1 reaction involves formation of a new or modified functional group or cleavage, oxidation, reduction, hydrolysis. These reactions are non-synthetic. Oxidation. This reaction involves addition of oxygen or negatively charged radical or removal of hydrogen or positively charged radical. Oxidation is the most important drug metabolism reaction. In many cases, the initial insertion of oxygen atom into the drug molecule produces short-lived, highly reactive quinine, epoxide, superoxide, E-intermediates, which then converted to more stable compounds. Oxidative reactions are mostly carried out by a group of monooxygenase in the liver, which is the final step involved a cytochrome P450 hemoprotein. NADPH cytochrome P450 reduces and O2. More than 100 cytochrome P450 isoenzyme differing in their affinity for various substrate or drugs have been identified. Barbiturates, phenothiazine, paracetamol, steroids, phenytoin, benzodiazepines, theophylline and many other drugs are oxidized by mitochondrial or cytoplasmic enzymes. Now we see about reduction. This reaction is converse or just opposite of oxidation and involves cytochrome P450 enzymes working in just the opposite direction. Drugs primarily reduced and chloral hydrate, chloramphenicol, halothiones. Then we see hydrolysis. This is cleavage of drug molecule by taking up a molecule of water. Ester hydrolyzed by esterase. Similarly, amides and polypeptides are hydrolyzed by amidase and peptidase. Hydrolysis occurs in liver, intestine, plasma and other tissues. Examples are choline esterase, procaine, lidocaine, procainamide, pethylidine, oxytocin. Now we see cyclization. This is the formation of ring structure from a straight chain compound. Example, proguanine. Decyclization. This is opening up of ring structure of the cyclic drug molecule. Example, barbiturates, phenytoin. This is generally a minor pathway. Phase 2. Reaction involves conjugation with an endogenous substance. Example, glucuronic acid, sulfate, glycine, etc. These reactions are synthetic. Conjugation reactions have high energy requirement. Glucuronide conjugation, this is the most important synthetic reaction. Compounds with the hydroxyl or carboxylic acid group are easily conjugated with glucuronic acid, which is derived from glucose. Examples are chloramphenicol, aspirin, p-henacetin, morphine, metronidazole. Other endogenous substrates like bilirubin, steroidal hormones and thyroxine utilizes this pathway. Acetylation. Compounds having amino or hydrazine residues are conjugated with the help of acetyl coenzyme A. Example, sulfonamides, isoniazids, PAS, hydrazine. Multiple gene control the acetyl transferase and rate of acetylation shows genetic polymorphism. Methylation. The amines and phenols can be methylated. Methionine and cysteine acting as methyl donors. Example, adrenaline, histamine, nicotinic acid. Sulfate conjugation. The phenolic compounds and steroids are sulfated by sulfokinase. Example, chloramphenicol, adrenal and other steroid. Glycine conjugation. Salicylates and other drugs having carboxylic acid group are conjugated with glycine. But this is not a major pathway of metabolism. Glutathione conjugation. Forming a mercapturate is normally a minor pathway. However, it serves to inactivate highly reactive quinine or epoxide intermediates forming during metabolism of certain drugs. Example, paracetamol. Ribonucleoside, nucleotide synthesis. It is important for the activation of many purine and pyrimidine antimethodoids used in cancer chemotherapy. Most drugs are metabolized by many pathways simultaneously or sequentially. As such, a variety of metabolites of a drug may be produced. Metabolites formed in synthetic reaction are more polar and thus more readily excreted by the kidney through urine and by liver through bile than those formed in non-synthetic reaction. First pass, presystemic metabolism. This refers to metabolism of a drug during its passage from the site of absorption into the systemic circulation. All orally administered drugs are exposed to drug metabolizing enzyme in the intestinal wall and liver. Presystemic metabolism of limited magnitude can also occur in the skin and in lungs. The extent of first pass metabolism differs for different drugs and is an important determinant of oral bioactivity. Now we come to another very important module of this topic, that is module 5, excretion. The kidneys are the principal organ for excreting water soluble substances. Generally, the contribution of intestine, saliva, sweat, breast milk and lungs to excretion is small except for exhalation of volatile anesthetic. Excretion via breast milk may affect the breastfeeding infant. Hepatic metabolism often increases drug polarity and water solubility. The resulting metabolites are then more readily excreted. Renal excretion. Renal filtration accounts for most drug excretion. About 1/5 of the plasma reaching the glomerulus is filtered through pores in the glomerular epithelium. Nearly all water and most electrolytes are passively and actively reabsorbed from the renal tubules back into the circulation. However, polar compounds, which accounts for most drug metabolites, cannot diffuse back into the circulation and are excreted unless a specific transport mechanism exists for the reabsorption. Example, as for glucose, ascorbic acid or vitamin B. With aging, for example, at age of 80, suppose for example, clearance is typically reduced to half of what it was at younger age, or say for age of 30. The principle of transmember passage governs renal handling of drugs. Drugs bound to plasma protein remain in the circulation. Only unbound drug is contained in the glomerular filter. Unionized form of drugs and their metabolites tend to be reabsorbed readily from the tubular fluid. Urine pH, which varies generally from 4.5 to 8 on pH scale, may markedly affect drug reabsorption and excretion because urine pH determines the ionization state of a weak acid or base. Acidification of urine increases reabsorption and decreases excretion of weak acids, and in contrast decreases reabsorption of weak bases. Alkalization of urine has the opposite effect. In some cases of overdose, these principles are used to enhance the excretion of weak bases or acids. Example, urine is alkalized to enhance excretion of acetyl salicylic acid. The extent to which changes in urinary pH alters the rate of drug elimination depends on the contribution of the renal route to total elimination, the polarity of the unionized form and the molecule's degree of ionization. Active tubular secretion in the proximal tubule is important in the elimination of many drugs. The energy dependent process may be blocked by metabolic inhibitors. When drug concentration is high, secretory transportation can reach at upper limit transport maximum. Each substance has a characteristic transport maximum. Anions and cations are handled by separate transport mechanism. Normally, the anion system eliminates metabolites conjugated with glycine, sulfate or glucuronic acid. Anions compete with each other for secretion. This competition can be used therapeutically. Example, probenecid blocks that normally rapid tubular secretion of penicillin, resulting in higher plasma penicillin concentration for a longer time. In the cation transport system, cations or organic bases, example, pamipexole, dofetilide are secreted by renal tubules. This process can be inhibited by cimetidine, trimethoprim, prochlorperazine, megastol or ketoconazole. Biliary excretion. Some drugs and their metabolites are extensively excreted in bile because they are transported across the biliary epithelium against a concentration gradient. Active secretory transport is required. When plasma drug concentration are high, secretory transport may approach an upper limit, that is transport maximum. Substance with similar physicochemical properties may compete for excretion. Drugs with a molecular weight of greater than 300 grams per mole and with both polar and lipophilic groups are more likely to be excreted in bile. Smaller molecules are generally excreted only in negligible amounts. Conjugation, particularly with glucuronic acid, felicitates biliary excretion. In the enterohepatic cycle, a drug secreted in bile is reabsorbed into the circulation from the intestine. Biliary excretion eliminates substance from the body only to the extent that enterohepatic cycle is incomplete when some of the secreted drug is not reabsorbed from the intestine. Now we come to final of this lecture series, that is module 6, conclusion. The study about pharmacokinetics and pharmacodynamics and drug action shows that drug therapeutics depends upon the health, age, amount or quantity of drug and many more factors which we have discussed earlier. We also studied about the biotransformation of drug by phase 1 and phase 2 reaction, which also changes the action of drug. Some drugs causes adverse effect in the body while other causes toxic effect along with main drug action. Some drugs directly act on the tissue while some firstly convert itself into metabolites which acts on tissue, and some drugs lose their potency on conversion to their metabolites. With all these information here, we come to the end of today's lecture. Do keep in mind what we have discussed today. It's time for you all to do some self-study. This is Professor Devashish Bose signing off for today. If you want to learn more and enhance your knowledge, you may log on to our website for MCQs, quizzes and LORs at www.cec.nic.in. Till then, goodbye.